Current efforts to try to treat the disease acutely when patients first get to the hospital have not been very successful. So most of the efforts are focused on secondary mechanisms of injury, which really result from the release of hemoglobin and heme from red blood cells. And understanding how hemoglobin can actually be toxic to the brain has been an important organizing principle for developing therapeutics for this condition. I think what is very exciting is that we're really understanding how having blood cells lyse in the brain actually leads to the death of neurons. And what is a remarkable insight is that the cells don't die by what's called necrosis. They're not kind of passively murdered, but they actually undergo a programmed form of cell death called ferroptosis. The ferroptosis is an iron-dependent form of cell death, and it's remarkable because it suggests that when you have a red blood cell lyse in the brain, it actually triggers signaling pathways that lead the cells to die. And because there's a biology to that process, it has two important implications. One, we can develop drugs that target that signaling pathway to actually inhibit cell death. And the second thing that we can do is we can understand what are the counterattack mechanisms that the body develops to actually prevent the cells from dying. And that's really been a very, very fruitful line of research, is understanding how does the brain create resilience to this programmed form of cell death called ferroptosis. Well, right now, I think we're at the point where we've identified drugs, and we're doing the critical developmental steps to ultimately be able to test those in humans. But part of the advantage that we have is because hemorrhagic stroke is a rare disease, I think the opportunities for accelerating approval and testing, particularly in the neurocritical care unit, are significant.